Welcome back, everyone. We're diving deep into the world of quality assurance in the lab today. Where those tests we rely on so heavily get done.
Absolutely. It's all about those results and making sure they're accurate and reliable.
Yeah, exactly. We've got a stack of materials here. Quality control in chemistry, quality control 123, and a bunch more. We're going to uncover the secrets of making sure those lab results are dependable. Lives depend on this stuff.
You're absolutely right. It's easy to picture labs as these, pristine, perfectly precise places. But the reality is it's much more complicated. We deal with intricate instruments, the human factor, and this ongoing fight against errors.
It's like a constant battle against those tiny gremlins that can mess things up.
Exactly. And that's where quality assurance comes in, right? It's like a safety net, protecting patients and building trust in the whole system.
Okay, so let's break down the safety net. We keep hearing quality control and quality assurance. Are those just like fancy ways of saying the same thing?
Not quite. They're related, but different. Think of a quality control, or QC, as like the day-to-day stuff, calibrating those machines, checking the reagents are good, and double-checking those results.
So QC is like the nuts and bolts of making sure each individual test is done right.
Yeah, exactly. And then there's QA, quality assurance. That's the bigger picture. Setting standards, training the team, having systems in place so that everyone's working together towards accuracy.
So QC is making sure each cake is baked right, and QA is about having the whole bakery run like a well-oiled machine.
Perfect analogy. And one of the key parts of QC is what we call internal quality control, or IQC. This is where those statistics come in.
Hold on, statistics. Thought this was a lab, not a math class.
Haha. Well, remember those control materials we talked about?
You mean like those samples with known values? They run through the testing to see if everything's working right?
Precisely. They're like secret agents sent in to make sure everything is on track. By analyzing how their results compare to what we expect, labs can keep an eye on their instruments, reagents, and, overall performance.
That's actually pretty clever. But how do they track all this data? Is there some super secret lab software?
Not quite secret software, but they do use a tool called the Levy-Jennings chart, LJ chart for short.
Yeah.
Like a graph, where each point represents a control result. It helps labs see trends or inconsistencies that might indicate a problem.
So if a point on the chart suddenly spikes or dips, that's a red flag, right?
Exactly. Now, those acceptable limits you see on an LJ chart, those are defined by something called standard deviations, or SDs.
Yeah.
They're like guardrails around the expected values. And if a result falls outside those guardrails, then it triggers further investigation.
Makes sense. So it's about setting those boundaries and knowing when something's off.
Right. And, sometimes rejecting those results outside 2 standard deviations can actually mean rejecting perfectly good runs.
Wait, what? Why would a lab reject good results? That seems kind of counterproductive.
It is about finding that balance, though, right? Think of those guardrails as a security checkpoint. Too wide and errors slip through. Too tight, and you get unnecessary delays and retests, even if the process is actually fine.
So it's about being cautious, but not overly cautious. But how do they decide when to investigate further and when to trust those results? Do they just have some sort of lab intuition?
Intuition helps, but they rely on a set of rules. Believe it or not, they're called the Westgard rules. It's almost like a cheat sheet for understanding those patterns on the LJ chart. Each rule represents a specific situation, and it helps pinpoint the kind of error they might be dealing with.
Okay, so if the LJ chart is like a map, the Westgard rules are the compass that guides us through the quality control landscape.
That's a great way to put it. Now, let's say a control result does go out of range. What happened then?
I'm hoping it's not panic in the lab.
No panic.
Yeah.
They have a systematic way to troubleshoot, a helpful flowchart. It's like, step by step, starting with the basics.
Okay, like making sure you didn't accidentally put salt instead of sugar in the cake batter. What if those initial checks don't find the problem?
Then they dig deeper. They check the calibration of the instrument, look for any system malfunctions, and if all else fails, they call in the experts, the service technicians.
So it's kind of like detective work, piecing together clues and using those Westgard rules to narrow down the possibilities.
Absolutely. You've got to find the culprit behind those out-of-range results.
Love the detective analogy. Speaking of culprits, what are some common causes of errors in the lab?
You've got 2 main types, random and systematic. Random errors are those unpredictable ones that pop up now and then, maybe a bubble in the reagent, a slight temperature change, even just a tiny variation in how someone pipetted a sample.
Those sound tough to catch. What about systematic errors?
Those point to a consistent problem. It could be a faulty calibration, a degraded region, even a slight variation in procedure that throws things off.
So random errors are like those occasional typos that slip through, but systematic errors are more like having a broken key on your keyboard that keeps typing the wrong letter.
Exactly. Identifying those systematic errors is crucial because they can lead to biased results across the board.
Now, the source mentioned third-party controls. What are those all about?
Think of them as like independent auditors coming from a different company than the lab's usual supplies. They help ensure those results are accurate and comparable to other labs, no matter what specific instruments or reagents they're using.
It's like getting a second opinion from a specialist, right? And I noticed they often have a longer shelf life. Why is that?
That's a great observation. Their longer shelf life means labs can use them as a consistent benchmark, even when switching to new batches of reagents or calibrators. Helps them detect any shifts in performance that might happen because of those changes.
So it's all about staying consistent and catching those tiny variations. Which brings us to lot-to-lot verification. Can you explain what that is?
Imagine baking a cake. You always use a specific brand of flour. One day they're out of your usual brand, you got to buy a different one. Lot-to-lot verification is like doing a test bake with the new flour to make sure it doesn't change your cake.
I love that analogy. So labs do this verification they get a new batch of controls, calibrators, or reagents.
Exactly. Another way to make sure those tiny changes in supplies don't lead to big changes in test results. And speaking of supplies, let's talk about those quality control materials, those QCMs. The source mentions different types, statistically and in terms of preparation. What are we dealing with here?
Okay, so statistically, you've got two main types, assayed and unassayed. Assayed QCMs, those come with predetermined values, like a pre-made cake batter, ready to go. Unassayed QCMs are more like a cake mix, where you add your own ingredients in this case, establishing the values in-house.
Got it. So it's like choosing between pre-made batter and a mix.
Right. And then for preparation, there are liquid or frozen QCMs, ready to use, and lyophilized QCMs. Those need some prep before you can use them. Okay, so options for both the stats side and the prep side. And it sounds like there are special handling instructions for some of these materials.
Absolutely. Some are sensitive to light, like bilirubin, while others are volatile, like ammonia. They can evaporate easily. Got to treat them carefully to get those accurate results. It all comes down to that attention to detail in the lab.
Fascinating how even those seemingly small things can have a big impact. Speaking of details, the source talks about parallel testing. When do labs do this?
Parallel testing is like a dress rehearsal. Imagine a theater company getting new costumes. You wouldn't wait till opening night to see how they look and fit on stage, right?
That would be a disaster. So parallel testing is like a trial run.
Precisely. When a lab gets a new batch of control material or makes big changes to its instruments, they run the new stuff alongside the old stuff to make sure they're getting similar results.
It's all about making sure those changes aren't causing any unexpected variations, right?
You got it. And they need a good amount of data for this. At least 20 data points collected over 10 days, and more is always better.
So the more data, the more confident they can be about those comparisons. Makes sense. And I see the source stresses using a stable analytical system before doing this parallel testing. Why is that so important?
Imagine trying to measure ingredients for a cake while the kitchen's shaking during an earthquake. You wouldn't get a very reliable measurement.
That's a great analogy. So if the lab system itself isn't stable, it's going to skew those results, right?
Exactly. You need that solid baseline to ensure you're making a fair and accurate comparison.
Okay, so before we jump into the next part of our deep dive, let's touch on those calculations mentioned in quality control and chemistry. I saw formulas for mean, standard deviation, and coefficient of variation. Pretty technical. Can you break it down for us non-math whizzes?
Sure. Think of these calculations as tools that help labs understand what their data is telling them. The mean, or average, gives you a central value. Standard deviation tells you how spread out the data is. Are the results close together, or are they all over the place? And then you've got the coefficient of variation, CV, that expresses that spread as a percentage of the mean.
So a lower CV basically means the results are more consistent, more reliable.
Yeah, that's it. Like aiming for a bullseye. The closer those areas are grouped, the better your aim is.
That's a fantastic visual. So we've explored the basics of quality control, dived into those LJ charts and Westgard rules, and even uncovered the secrets of those third-party controls and parallel testing. It's incredible how much work goes into making sure those test results we rely on are accurate and dependable.
And we've only just scratched the surface. There's so much more to discover about quality assurance.
Well, stay tuned, everyone, because we're about to dive even deeper. It really is. It's like peeling back the layers of an onion, more complexity and details with each layer.
Exactly. And each layer contributes to that ultimate goal, right? Ensuring patients get accurate and reliable results. Those results, they can be life-changing, diagnosing a condition, guiding treatment, impacting someone's whole health journey.
It really underscores the responsibility labs carry. They're not just processing samples, they're generating data that directly impacts people's lives.
Absolutely. Speaking of responsibility, the source, quality control in chemistry, mentions these built-in controls. What are those all about? Is it like a tiny quality control expert living inside each machine?
Now that would be cool, but I'm guessing it's a bit more technical than that.
You got it. Built-in controls are basically automatic checks, like integrated right into certain testing systems. They might run with each test or at specific times, constantly monitoring how the instrument's doing.
So it's like having a spell checker in your word processor, catching those typos before you even see them.
Exactly. And these built-in controls, they're especially important for tests that give critical information, like blood gas analyzers. Those measure things like pH, oxygen, and carbon dioxide levels in blood samples. Information doctors need right away to make those life-saving decisions. Having those built-in checks just adds another layer of confidence in those results.
That makes a lot of sense. It's reassuring to know those extra safeguards are there, especially when time is so critical.
Absolutely. And remember how we were talking about QC being the daily grind and QA being the bigger picture? Well, those monthly QC reports mentioned in quality control and chemistry, those are like a progress report,
Yeah.
showing how well the lab is keeping up those standards.
So if the daily QC tasks are like keeping the kitchen clean, these monthly reports are like doing a deep clean and inspection.
That's a great way to put it. They basically summarize all that quality control data from the whole month, including those metrics we talked about, the mean, standard deviation, and CV for each test.
So it helps them spot any trends, figure out where they can improve, and basically make sure they're consistently hitting those quality targets.
Precisely, like checking your car's engine regularly to make sure everything's running smoothly.
Yeah.
Catch those potential issues before they turn into big problems.
And speaking of potential problems, let's go back to those two main types of errors we were discussing earlier, those systematic and random errors. Can you remind us what makes them different, and why is it so important to be able to tell them apart?
Okay, so think of systematic errors as those pesky gremlins that always push your results in one direction. It's like a clock that's always 5 minutes fast, consistently off, leading to those biased results.
So it's like a recipe where you always add a bit too much salt, that dish will always taste a bit off.
Exactly. And those errors often point to a specific issue that needs fixing, like a faulty calibration or a reagent that's gone bad.
Got it. So what about random errors? Are they as easy to pinpoint and fix?
Random errors are more like, those typos that sneak into your writing. Unpredictable, and often caused by those tiny variations in the testing process. A tiny bubble in the reagent, a little temperature change, even just the inherent imprecision of some methods.
So systematic errors are like a consistent bias, while random errors are like fluctuations that are harder to predict.
Exactly. Both need to be carefully watched, though, because they can affect the accuracy of those test results.
And that's where our trusty Levy-Jennings chart comes back in, right? It helps labs visualize those patterns and spot those errors, whether they're those systematic shifts or those random blips.
You got it. The LJ chart is like that detective's whiteboard, allowing labs to plot the clues and track those errors over time. Speaking of visuals, quality control in chemistry brings up the Gaussian distribution, the normal distribution. You might know it as the bell curve.
Ah, yes, the bell curve. I remember that from stats class, but it's all a bit fuzzy now. Can you refresh our memory? What is it, and why does it matter for quality control?
Imagine lining up everyone in a city by height. You'd see most people clustered around an average height, right?
Oh.
With fewer people at the extremes, the very tall and the very short. That's essentially the Gaussian distribution, that natural tendency for data to cluster around a central value, and those outliers becoming less frequent as you move away from the average.
So it's like a visual way of showing how data naturally spreads out. And I vaguely remember those standard deviations we talked about earlier being part of this bell curve.
You're right. Those standard deviations, they're like markers on that bell curve, defining those ranges of probability. They tell us what percentage of data points we can expect to find within a certain distance from that average.
Okay, so it's not just about the average. It's also about understanding how spread out the data is around that average. How does this all connect back to the Levy-Jennings charts and quality control in the lab?
Well, the LJ chart is built on the idea that quality control data follows a Gaussian distribution. This allows labs to use those standard deviation markers to set those acceptable limits for control results. So if a result falls outside those limits, too far from the average, it triggers that investigation.
So the bell curve provides that framework for understanding the expected spread of results, and the LJ chart helps visualize any deviations from that expected pattern.
Exactly. And remember that troubleshooting flow chart? Well, quality control in chemistry goes even deeper, outlining that systematic approach to investigating those out-of-range results.
It's like a choose-your-own-adventure for lab professionals. So if a lab gets a control result that's outside those acceptable limits, they follow this flow chart, step by step, to figure out what's causing the problem.
Exactly. The flowchart starts by asking, are multiple tests affected, or just one? If it's multiple, it might be a system-wide issue, like a calibration problem affecting everything. If it's just one test, then they'll start with those basic checks. Control prep, region handling, using fresh materials and all that.
So it's a process of elimination, right?
Sure.
Rule out the common causes first and then dig deeper if they need to.
Precisely. This helps ensure they're tackling the root of the problem, not just masking the symptoms, like, going to the doctor with a cough. They won't just give you cough syrup. They'll try to figure out what's causing it.
That's a great analogy. It shows how important it is to not just jump to conclusions, but really take that systematic approach to troubleshooting.
Definitely. Now, how we talked about common error causes, like those bubbles in reagents, faulty calibrations? Well, there are some sneakiest culprits, those that fly under the radar.
Oh, like what? Tell me about these hidden gremlins.
Well, one often overlooked factor is sample integrity. If a blood sample isn't collected or handled properly, it can really throw off those results, no matter how good the lab's procedures are.
So it's not just about what happens inside the lab. It's about everything that happens before the sample even gets there.
Exactly. That pre-analytical phase, the blood draw, transportation, storage, all of it can have a huge impact. For example, if a blood sample gets hemolyzed, where the red blood cells rupture, it can interfere with certain tests.
It's like trying to bake a cake with rotten eggs. No matter how good the recipe or the oven is, that cake's not going to turn out right.
Perfect analogy. It just highlights how everything's connected in lab testing, from the patient to the doctor to the lab and back again.
It's a chain of custody, and every link needs to be strong for those results to be accurate and ultimately for the patient to be safe. Speaking of integrity, let's talk about those expiration dates and stability dates the source mentions. Might seem small, but it's crucial for quality and reliability.
Right.
Okay, I've got to admit, I've always been confused by the difference between those dates. Aren't they basically the same thing?
Not quite. Think of it like this. The expiration date is the manufacturer's guarantee, like the best before date on food. It's the last date they can guarantee the product's quality. The stability date, on the other hand, well, that comes in after a solution's opened or prepared. It's figured out by the lab staff, based on the manufacturer's recommendations, and it shows how long that solution stays stable and reliable for testing.
So the expiration date's like the best before on a milk carton, and the stability date is like that mental note you make to use the milk within a week of opening.
That's a great way to remember it. And just like you wouldn't want to drink spoiled milk, labs don't want to use expired or unstable solutions for testing. It's all about keeping those materials good to make sure those results are accurate.
And I see the source highlights proper labeling, clearly marking everything with the name and date of opening.
Absolutely. Proper labeling is like a passport for each solution, letting staff track its journey and make sure that only those fresh, reliable materials are used. It's all about traceability and accountability.
It's incredible how much thought goes into every single detail. We've covered so much built-in controls, monthly QC reports, the differences between those systematic and random errors, and the importance of sample integrity and labeling.
And even touched on the world of stats again.
It's been a fascinating journey so far. I can't wait to see what other insights await us as we continue to explore. It's amazing how everything connects to make sure those lab results are top-notch. But it's not just about, abstract ideas and procedures, is it? It's about real people and their health.
Absolutely. Every single point on that LJ chart, those rule violations, each troubleshooting step, it all comes back to the patients. Their safety and well-being. We can't forget that.
You're so right. It's about making sure people get the right diagnoses and the right treatments. Speaking of comparing, the source mentioned this coefficient of variation ratio, CVR. What's that all about?
Think of CVR as a way for labs to, see how their precision measures up against others.
Yeah.
Like a friendly competition to see who's getting those consistent results.
So it's like those baking shows where they judge you on taste A&D, how it looks. You need that balance of flavor and, like, precision.
Exactly. A lower CVR usually means better precision compared to other labs doing the same test. The source even gave this example, a lab with a CVR of 0.97 for iron testing. They're actually doing a bit better than average.
It's like getting an A+ for precision. But what's the real-world impact? Why should patients care about a lab's CVR?
Well, imagine you're a doctor using those iron results to, say, monitor a patient. Knowing that the lab has a low CVR gives you more confidence in those numbers, right? Helps you make those informed treatment decisions.
It's like knowing the thermometer you use to check your kid's temperature is accurate. You wouldn't want to make a decision based on a wrong reading.
Exactly. It all goes back to trust in that information. That's what guides those healthcare decisions.
Okay, so as we wrap up our deep dive into this world of quality assurance, what's the one key message you hope our listeners take away?
It's more than just ticking boxes and following those procedures. Quality assurance in a lab is about making sure every result is as accurate as it can be. Because at the end of the day, it's about protecting those patients and giving them the best possible chance for a good outcome.
I love that. It's like we've been behind the scenes uncovering all those safeguards that make sure those results are reliable. And it's something we often take for granted.
Yeah, I hope this deep dive has shown just how much work and dedication goes into making sure those results are trustworthy. The people in those labs, they're working tirelessly.
To our listeners, remember those lab reports? They're not just pieces of paper. They represent this complex process designed to protect your health. And if you're ever curious about a test or result, just ask your doctor. Don't be shy.
Definitely. Knowledge is power, right? Understanding the world of lab testing, it can help you be a better advocate for your own health.
Thanks for joining us on this deep dive. We've got more exciting topics coming up, so stay tuned. Until then, stay curious and stay well.